So we are continuing on with our appendix lectures, just digging a little bit deeper into some interesting odds and ends in the chemistry of DNA. Once again, with a view to looking at the issues that suggest fairly deliberate design. And the issue we will tackle in this short mini lecture is why ribose as the sugar for DNA and RNA, and as you'll notice, I'm using ribose generically as the specific five-membered sugar. Now, this is a topic which has interested chemists, and if we simply restrict ourselves to the five-carbon sugars, although there's no particular reason to do that, it is interesting that even then there would have been a very early choice that had to be made between the 8 pentose sugars that you see here on the slide. There are 2 ribose enantiomers, 2 arabinose enantiomers, 2 xylose enantiomers, and 2 lixose enantiomers. So between the D and L configurations, there would have been 8 possible pentose sugars for the genetic code to choose to use in the DNA/RNA backbone. And it is a very interesting question as to why ribose in particular, and even more specifically D-ribose, was chosen. For those interested in a deeper dive, let me refer you to this paper, Why Ribose Was Selected as the Sugar Component of Nucleic Acids from DNA and Cell Biology back in 2006. And this was a theoretical paper looking at a variety of different calculations about what the different possible pentose sugars could do or would do to the structure of DNA. And their conclusion that you see on the fourth line is that ribose was not randomly selected, but the only choice, since beta-D ribose fits best into the structure of physiological forms of nucleic acids. And they looked at arabinose, xylose, lixose, and so forth, and found that the hydroxyl groups in the C2 carbon or C3 carbon are above the so-called furanose ring and will cause steric interference with the C5 hydroxyl group. So basically, of the 8 possible enantiomers, only the one that works was chosen. And the question is, how is it that nature was able to make this choice so early on? And there is, in fact, a very rich chemical synthetic literature on xeno-DNA. XENO, xeno-DNA, or forgive me, I should say xenonucleic acids rather than xeno-DNA, xenonucleic acids, where synthetic chemists have used sugars other than D-ribose to synthesize nucleic acids. And in general, although there's a fairly rich literature that would seem to support that, yes, this could be done, a closer look tends to suggest that no, actually it couldn't, and a variety of reasons pop up. Number one, the helical structure, the double helix structure of DNA cannot be supported by most other sugars. A few of them can conceivably make a stable double helix. Number two, the required spacing of 25 angstroms to allow one double cyclic nucleic acid and one single cyclic nucleic acid, such as adenine and thymine, to hook together, That spacing requires ribose. Thirdly, DNA polymerase to copy and to synthesize DNA cannot read xenonucleic acids. So we will see what the future brings in this synthetic arena, but for now it certainly seems ribose is by far the best choice. And for those of you who are truly interested in chemistry, I refer you to this paper by Professor James Tour at Rice University called Animed Versions of a Synthetic Chemist. It is about micro machines that they synthesize, but the end of the paper goes into a discussion of how unusual it is that ribose could have been synthesized in the prebiotic environment, because it is very difficult to synthesize, number one. Number two, it would be associated with a lot of other polycyclic carbon type sugars. We could call those impurities if we're just looking for simply D-ribose. And over time, the D-ribose that we want tends to degrade, and these other impurities are byproducts of any reasonable scenario of prebiotic synthesis tend to increase. So how is it that DNA was able to fixate on this ribose sugar truly remains a mystery when it is compared to other saccharides. And now finally, a point that really should not be overlooked, and one that I find also truly amazing. I've been talking about ribose generically for DNA and RNA, and I'm sure some of you have already sort of raised an eyebrow because you know that DNA uses deoxyribose, and that's why it is called DNA, deoxyribonucleic acid. So the D is for the deoxyribose component, and RNA is ribonucleic acid, and so RNA uses ribose. And here you have the structure of deoxyribose on your left and the structure of ribose on your right. Deoxyribose used in DNA, ribose used in RNA. And you can see that they are both a 5-carbon sugar ring. One of the members of that ring is oxygen. The 5th carbon hangs above the ring. And the big difference in the structure is in the 2 carbon, the number 2 carbon, where ribose has a hydroxyl group, but deoxyribose has that oxygen atom taken out. And that is why it is called deoxyribose. The difference is in the 2 carbon position, a hydroxyl group in ribose, and no hydroxyl group, but just a hydrogen atom in deoxyribose. Now, what is the big difference? What is the big deal? Well, remember we talked about how stable phosphodiester bonds are, and that this stability allows for the remarkable stability of genetic material, and it also allows for selective hydrolysis. Now, what we want is for DNA, which is kind of the hard drive of genetic information, to be very, very stable, but RNA, which is a copy or template made from DNA for the temporary synthesis of proteins, to be more quickly degradable. Because otherwise, once a messenger RNA is copied from DNA, it would keep hanging out and making protein. And we don't want that. We want to make the protein when it is needed, and then that mRNA needs to be degraded. And it turns out that that is precisely the difference that this hydroxyl group in ribose makes. So you can see from Professor Westheimer's paper that we referred to earlier, Why Nature Chose Phosphates, the presence of a hydroxyl group at the 2 carbon allows the formation of a 2 prime 3' cyclic phosphate compound to be made that allows for cleavage of RNA, hydrolysis of RNA, and the difference is that RNA can be hydrolyzed and cleaved and broken down 100 times faster than DNA, precisely because of the presence of that hydroxyl group on the 2 carbon that can allow it to react with a phosphate anion to come in and break that RNA chain. And so it is really quite remarkable that all of these things came into being simultaneously, because they all had to be there pretty much simultaneously together to allow the system to work. DNA be stable, DNA get turned into RNA, RNA be relatively stable but much more easily hydrolyzed, and so on. And when one looks at the entire chemical synthetic picture, it is utterly remarkable that this system came into being. So I hope that you enjoyed this part of the lecture, and we probably have just one more small excursion, once again for those who love chemistry, and then that will bring our entire series, including the appendix lectures, to an end. Thank you. Assalamu alaikum.